All right, we are going to cover chapter 26 today on population genetics. So this is, I think, a very interesting chapter that kind of takes all of the material that you've already learned and puts it together to have some really cool practical applications that we're going to go through. So these are the key concepts and the material that we are going to cover. And you can go through these later once you've gone through the entire lecture. But kind of a broad overview is we're going to take genetics and now we're going to apply it to human populations in order to make some really cool research observations and also have some really cool practical applications. And so we've seen some of these practical applications before. So in the first lecture of the class, I covered some of the basics of 23andMe. So I've done 23andMe, which is a company that takes a saliva sample and uses that saliva sample to genotype a large number of alleles that exist within our cells. And it uses that to infer things like ancestry. So I have my ancestry report that reports I'm 99.9% European, and then it also breaks down this European ancestry into some of these broad geographical categories like British and Irish, specifically Ireland, French and German, I guess specifically Netherlands, and then Scandinavian and kind of these other categories. So we're going to cover in this lecture how this is possible using information that's encoded in our DNA. We've also seen how you can identify close relatives. So this is a completely different algorithm that 23andMe uses, but we can use the information from the previous chapter 5 on linkage in order to identify close ancestors. And by close ancestors, I mean things like brothers, sisters, fathers, mothers, cousins, aunts, uncles, second to third cousins, etc. So once you get a little bit past this like second to third cousin ancestry, then it becomes more and more difficult to actually identify these close relatives. But again, this is a real life example. So this is from my 23andMe report, and so I can see information that I share with my brother. So this is the 23 chromosomes that my brother and I share. And you can see which parts of the chromosomes are identical between us. So again, we have the same father and mother, and so we can inherit identical or different chromosomes from them. And so the gray indicates that neither part of this chromosome is the same. The light purple is that one of the chromosomes is the same, and then the dark purple is both homologous chromosomes are the same. And so you can see the closer the relationship, the more similar your DNA is. And then when we get to like a second and third cousin, you can see that there's very little DNA that is actually shared. So we only have this second to third cousin that I don't know, I've never met, but we only have 6 small regions of the chromosome that we share at all. But because of this, we're actually able to identify ourselves as relations. All right, and so this is possible because we are not identical to each other. So each person in this room has 2 chromosomes, and these chromosomes are different from each other, and they're different from the human reference genome. So on average, there's about 1 in every 1,000 nucleotides that is actually different. So if we can't compare the sequence of my chromosome 1 from my mother, and we compare it to the DNA reference, on average, 1 out of 1,000 nucleotides would be different. But because there is so much DNA, there's 3 gigabases of DNA within our cells, within a haploid cell, that means that each person has about 3 million differences from the reference. And so there's a huge amount of DNA differences. These can translate to things like phenotypic differences, so kind of why our hair is different color, why our eyes are different color, but it can also just have no effect on our phenotype. So most of these genetic variations actually are thought to have almost no effect on our phenotypes at all. But they do encode information about, for example, where we're from and who we're related to. So this chapter, part of this chapter is kind of getting into these polymorphisms, and one of the parts about these polymorphisms is that this explains kind of why we have done what we have done with making homologous chromosomes different colors and kind of what the idea is behind that. So, if we had a cell, I've always encouraged you to distinguish the chromosome from your father and the homologous chromosome from your mother with different colors. And pretty much how it works is that any unrelated chromosome, so if you also compare your chromosome to someone else in the room, as long as you're unrelated to that person, they also should have a completely different color. And so where does this kind of color scheme, where does the idea behind this come from? And that is from actually analyzing these alleles and these polymorphisms on all these homologous chromosomes. So here I'm showing you, so you can think about this as like your chromosome 1 from your father. And so if we look at this chromosome 1 and we identify all of the differences from the reference, we can mark that in red. And so this position right here, instead of being in G, is actually an A nucleotide. And if we look at our mothers, for example, her chromosome matches the reference. And then if you look at, a friend in the room, one of their chromosomes matches the reference, and one of them is the alternative allele. And then if you look at a fifth one, this would be A. And we can go through and do this for the entire chromosome and identify all of the differences. And so we can see that, right here we've listed 9 of these alternative sites that differ from the reference. And then we've also gone in and looked to see what the nucleotide is at these other 5 chromosomes. This is just a schematic, but in reality there would be approximately 200,000 differences on a single chromosome. So we would do this and list 200,000 letters. And so what you see is that each one of these chromosomes has their own unique combination. And so, A and C are not always matched to each other. You can have an A matched with a C, or you could, for example, have an A matched with a T, or a G matched with a C. And so all of these polymorphisms are independent from each other. And so what you have is a unique combination. So, this chromosome right here has 2 of the 9 differences from the reference. This has 3, this has 4, this has 4, this has 4. But every chromosome has its own unique combination of things that are different. And so this is kind of why we color code these, is because each of these chromosomes acts completely independent of each other. And so this is the start of our analysis, both in terms of linkage analysis, but now we can start to get into this idea of allele frequencies as well. And so in this lecture, we're going to cover populations, and so what exactly a population is, and then some of the important measures of that are allele frequency and then genotype frequency within that population. We're going to talk about Hardy-Weinberg, and so this is just a mathematical approach that allows us to predict the genotype frequency from the allele frequency. We're going to talk about evolutionary forces that change allele frequencies. So evolution, the way it acts is that it changes the frequency of something in the population. So we'll talk about things like natural selection, genetic drift, and some real-life examples of those. And then finally, we'll talk about how we can use linkage to infer ancestry.